Greetings everyone, and welcome to this new Gen webinar entitled Reporter Virus Tools to Combat Current Viral Threats and the Next Pandemic. This webinar was made possible through sponsorship from Integral Molecular. I'll be your host today. I'm Jeff Bogaliska's technical editor for Gen, which has been at the forefront of the life sciences industry for over 40 years. Providing you the latest news and insights on the newest tools and technologies. Now, prior to the COVID-19 pandemic, investigators not actively working in the field of virology probably didn't give it much thought. Well, that paradigm has changed considerably over the past 3 years as the pandemic brought virology to the forefront of many research programs. But now, with more groups than ever devoting time to virology research, a very large question still lingers. How do we get ahead of the next viral threat? And this perplexing question is compounded by the fact that there is a very limited number of commercial labs that are licensed to conduct studies using BSL-3 and BSL-4 pathogens. Well, thankfully, tools do exist, such as ready-to-use reporter virus particles, or RVPs, that have enabled virus research in BSL-2 environments, allowing for the testing of sera and antibodies in high throughput, along with safe neutralization assays to accelerate virology research. Now, this will all be the center point of our presentation today and what our speakers are going to tell us much more about in just a few moments. But first, let's meet our guest for today's event and get to know them a bit better. Dr. Hayley Crawford will lead things off today. Hayley is a virology research and application scientist at Integral Molecular and works on the company's extensive RVP portfolio. Hayley has vast experience in virology and immunology, which allows her to work with customers directly to help them develop the right solutions for their research endeavors. And today, she'll discuss Integral Molecular's broad catalog of RVPs, including influenza A and B, Marburg virus, and the latest variants of SARS-CoV-2, in addition to discussing the company's expertise in custom RVP projects. Hayley will then turn the presentation over to Dr. Sheila Keating, who is the Vice President of Immunology at Gigagen, a San Francisco-based subsidiary of Grifols that is advancing antibody drugs for a host of diseases using single-cell technology. Sheila has almost 20 years of industry experience and is an author on more than 100 scientific publications. And today, Sheila is going to tell us about how Gigagen utilized pseudoviral neutralization assays to develop an antibody therapeutic against SARS-CoV-2. Then Sheila will turn things over to our final presenter for today, Dr. Natalie Anosova, who is Director of Immunology and Head of Preclinical Immunobiology at Sanofi. Natalie has an immensely strong background in immunology with more than 6 years of experience in academia and over 19 years in industry R&D, 18 of those being spent at Sanofi Vaccines. Natalie has worked tirelessly in the development of immunological in vitro and in vivo models for identification and validation of viral and bacterial targets for therapeutic and vaccine projects, enabling progression to the clinic. And today, she's going to describe her team's experience working with RVPs in the development of a vaccine candidate for SARS-CoV-2. Now, before we get the webinar underway, though, I do want to remind the audience to stick around for our Q&A session that will begin right after the presentation ends. All of our presenters are going to be on hand to try to answer as many of your questions as possible. So, if you have any questions for Hayley, Natalie, or Sheila, all you need to do is type your question into the Ask a Question box and hit submit. Now, you can do this at any time during the presentation. You don't have to wait until the very end. So if a thought pops in your head and you'd like to address it to one of the speakers, go ahead and type it in and hit submit. Okay, with that said, I'm going to turn things over to Hayley to get things started.
Hello, everyone. Thank you for joining us to find out how our reporter virus tools can be used to combat current viral threats and the next pandemic. I'm Hayley Crawford from Integral Molecular. We are a biotechnology company founded by virologists in Philadelphia. Deep expertise in virology is at the core of our 20-year history. Our technologies and R&D services have helped over 400 companies working in vaccine research and drug discovery, and we have authored over 350 peer-reviewed papers in journals including Cell, Science, and Nature. And we deeply value all of these collaborations. Our reporter-virus particles, or RVPs, are replication-incompetent, pseudotyped virus particles that enable safe BSL-2, easy and high-throughput viral infectivity and neutralization assays using standard detection instrumentation. RVPs display antigenically correct spike protein on a heterologous viral core, and they carry a modified genome with a reporter gene for quantitative readout. On the bottom left, there's a nice visualization of our RVPs in these pseudo-colored scanning electron microscopy images from our customers at the University of Chicago. They showed our SARS-CoV-2 RVPs in green, bound to their proprietary nanotraps, coated with ACE2 protein. Other applications include use in vaccine trials, monoclonal antibody lot release, sera screening, and so on. Over the past 10 years, scientists at Integral Molecular have been on the forefront of combating viral epidemics such as Zika, Ebola, and chikungunya, in addition to working on Marburg, VSV, dengue, SARS-1, MERS, and over 70 CoV-2 variant viruses. We produce RVPs with an optical reporter gene, either luciferase on the left or GFP in the middle, that is expressed within 24 hours of cellular infection by the RVPs. Readout for luciferase is in relative light units and in percentage of infected cells for GFP. The dose response curves on the right show how RVPs are used to characterize the neutralization of monoclonal antibodies and sera from patients. Our RVPs are available as a ready-to-use reagent that provides a safe and efficient alternative to the historical standard plaque reduction neutralization test, or PRINT. The left figure is from one of our valued customers at the University of Pittsburgh, and other customers of ours have published similar data. We've also shown that different RVP production lots are very comparable, and that results are reproducible across different RVP inputs for a given well or experiment, because we know that having a reliable reagent is essential to our customers so they can trust their results. The huge variety in our RVP catalog enables neutralization profiling for monoclonal antibodies or immune responses in patient or vaccinee sera. For example, testing a monoclonal antibody and serum from a customer, we found that while the SARS-CoV-2 reference strain in red was neutralized, the variants in blue were not strongly neutralized. Our customer Immunome tested a large panel of different RVP variants, and their 3 antibody cocktail was able to neutralize them all. This profiling is very important for insights into vaccine responses and therapeutic antibody efficacy. Our RVPs are produced under quality-controlled conditions as a critical reagent to enable regulatory submissions. Dr. Anosova from Sanofi will later tell us how she used RVPs for her vaccine development. And before that, Dr. Keating from Gigagen will describe her use of our RVPs to develop and screen antibodies. And this FDA information is available on our website too. We have the most extensive catalog of pseudoviral particles, especially when it comes to SARS-CoV-2, which has now infected over 600 million people worldwide. Keeping up with its rapid evolution has allowed us to produce RVPs soon after a new variant is identified, and this enables our customers to keep up with the ever-changing landscape of infections and treatment efficacies. We expanded to produce SARS-1 and MERS, and recently added influenza and filoviruses to our catalog that has long included dengue and Zika. We can also develop custom RVPs by request and have been able to advance many customer projects this way. Our customers at Takeda used our Zika RVPs to measure immune responses in their phase 1 vaccine trial. They developed a robust automatable neutralization assay, and in 271 volunteers, they tested serum over 3 time points. Their data shows that the neutralizing antibody titers measured were comparable between RVPs and PRINT across different vaccine doses. This shows the value of using RVPs to characterize immunological endpoints in a vaccine clinical trial. And in addition to their use in a clinical trial setting, we have published a peer-reviewed paper demonstrating Zika RVP's key properties as a critical reagent, including stability and lot-to-lot consistency. In another trial by Takeda, on their recently approved dengue vaccine, our dengue RVPs were used to screen for and measure antibody responses in vaccinee sera. This live attenuated tetravalent vaccine consisted of dengue 2 and 3 recombinant viruses, as well as dengue 1, 3, and 4 surface proteins. They measured a strong dengue 2 response and wanted to ensure that there were responses to the other serotypes too, which you can see in these 3 figures. From an efficacy standpoint for dengue, it's important to have responses to all of these other serotypes. And again, we previously published a paper describing the statistical agreement between live virus print results and the RVP assay, as well as the reproducibility and speed of dengue RVP assays. Moving on to the filoviruses, Ebola and Marburg. Philo means thread, and these thread-like viruses are 2 of the world's most deadly viruses. They are highly contagious and cause sporadic outbreaks of hemorrhagic fever. Transmission is from wild animals to humans and then human to human, and the mortality rate is incredibly high, between 60 and 90% for Ebola, depending on the strain, and around 50% for Marburg. There is currently an Ebola outbreak in Uganda that started in September this year, and a Marburg outbreak in Ghana just ended last month. A major obstacle for live virus research with these deadly, highly infectious filoviruses is that they require a BSL-4 lab and specialized equipment to ensure scientists' safety. We applied our virology expertise to these, and we now have off-the-shelf safe reagents in our catalog for these 2 viruses. Our Ebola and Marburg RVPs eliminate the need for growing your own virus and allow any researcher with a BSL-2 lab to work with them. They are very infectious while remaining safe, and every RVP in the vial is identical. There's little to no risk of antigenic shift or drift, which can occur when culturing live virus, and the safety of these pseudoviruses cannot be overemphasized. We have Marburg in GFP and luciferase, and Ebola in GFP. We recently added some influenza strains to our catalog. Flu types A and B are the 2 main types of influenza and are responsible for seasonal flu epidemics each year. Globally, these viruses infect 1 billion people and cause up to 650,000 deaths per year. Flu A has 2 surface proteins, hemagglutinin and neuraminidase. There are many subtypes of these genes, and combinations of these 2 give us the flu A strains. Birds are the primary reservoir, and these viruses can cause pandemics. For example, genetic reassortment caused the pandemic strain H1N1 in 2009 that we know as swine flu. Flu B has 2 lineages, Victoria and Yamagata. These viruses circulate only in humans. 2 A strains and 2 B strains are included in quadrivalent flu vaccines every winter. Looking first at the influenza A strains, surveillance and pandemic preparedness efforts are focused on H5 and H7 viruses, and we now have H5N1 RVPs available in our catalog in both GFP and luciferase. They are very infectious while remaining safe. The GFP RVPs infect almost 100% of target HEC cells, and luciferase RVPs require a very low volume per well to achieve a high signal to background value. We also have influenza B RVPs, both the Washington strain, which dominates the circulating strains, and Phuket, which is in the flu vaccine. These RVPs are easily scalable, genetically stable, and inherently safer than native viruses. Many of our RVP customers are interested in the epitopes that their antibodies target, and we use our shotgun mutagenesis libraries of viral surface proteins to elucidate these epitopes down to the single amino acid residues. We also offer a platform to profile antibody specificity and off-target safety in live cells. This is our membrane proteome array. And to conclude, our technologies support faster and safer vaccine and therapeutic antibody development against the world's deadliest viruses and enable the greater scientific community to ramp up studies of new viral threats as soon as they emerge. Thank you for your attention today, and thank you to my amazing team at Integral Molecular. And with that, I will hand over the mic to Dr. Sheila Keating from Gigagen.
Hi, my name is Sheila Keating. I'm the Vice President of Immunology at Gigagen, a subsidiary of Grifols. And today I'm going to talk to you about our pseudoviral neutralization assays and how we use them to develop our first-in-human recombinant polyclonal antibody therapeutic for SARS-CoV-2. So using our technology, we can capture the entire antibody repertoire by taking a blood sample, isolating B cells, and running them through our droplet capture system. And this allows us to recreate the entire antibody repertoire using recombinant technology. This will allow us to make polyclonal antibody therapeutics, or we can find very rare and exceptional monoclonal antibodies. So for our SARS-CoV-2 product, we used convalescent donors because we knew that they had antibodies that helped them recover from the infection. We were also able to use, if we needed to, we were also able to use rat or chicken immunoglobulin humanized animals. So after isolating the B cells from either the convalescent donor or a rat or a chicken immunized animal, we run those B cells through our droplet capture system. And we use single cell genomics that allows us to retain the linkage between the heavy and light chain by creating a single chain variable fragment. We take those fragments and we'll incorporate them into plasmid vectors for transfection, and we will transfect them into CHO cells, or we can take that sequence and we can express it on the surface of yeast. And that will allow us to find and sort to enrich for antibodies that are specific for any target of interest. And in the case of SARS-CoV-2, it was the receptor binding domain. So this process is extremely fast. We can take blood samples within a few weeks of infection, or we can do the immunizations, and it will take 1 to 2 months to build up the antibody response required. It takes only a few days to do that linkage between the heavy and light chain, a week to clone our cells into a plasmid ready for transfection, and then that selection process takes up to 4 weeks. And we can enrich for those antibodies that we're really interested in, and that takes 2 to 4 additional weeks. So altogether, this is about 2 to 3 months of work to get a final library that is ready to produce thousands of antibodies that will target your antigen of interest. So as I mentioned, we use yeast display to express the single chain variable fragment. And we use a fluorescently labeled antigen to bind to those antibodies that we're interested in identifying. And so during the first round of sorting, we may not have very many antibodies that are specific to the target of interest, such as the receptor binding domain, but we will go through 3 rounds of enrichment so that we will find all of the antibodies that are specific to the receptor binding domain. And we use deep sequencing as a quality control to identify what are the sequences that are binding to the antigens of interest. So next, we'll take those sequences and then we'll clone them into our vector, ready for transfection. So this is a proprietary and patented technology that we've developed, taking those single chain variable fragments and turning them into antibodies ready to be made by our CHO cells. One of the unique features is that it's a polyclonal antibody library, but we can clone in modifications to the Fc that will enable the antibodies to have better antibody-dependent functions. So either antibody-dependent cellular cytotoxicity, longer half-life, or even a modification that will block the ability of antibodies to bind and blocking the potential for antibody-dependent enhancement. Another really unique feature about our technology is that we're only getting one sequence of antibody into a plasmid, we only want one plasmid to be transfected into a CHO cell, so that each CHO cell can only produce one antibody. And so we have a site-directed system where we can incorporate a single plasmid into a single CHO cell. We've also demonstrated that we're able to get high yields in our antibody production. So at the end of this process, we'll have thousands of antibodies being produced by thousands of different CHO cells. And each CHO cell will have an antibody, a single antibody copy that's being produced. So this final product is very similar to the original repertoire of antibodies that we got from B cells. And once we're finished with our cloning, with our transfection, we will grow our antibodies in culture similar to a monoclonal antibody, and we can use the same process that's been used for purifying monoclonal antibodies. We take our reactors that we've grown, our CHO cells producing those antibodies. We'll harvest the cell culture fluid. We'll do a protein A affinity purification, cation exchange, anion exchange, ultrafiltration and diafiltration, and then our final product is very similar to what you would get after you've purified a monoclonal antibody. And so all of the existing manufacturing infrastructure that's used for monoclonals can also be used for polyclonal. So we were very lucky to find the pseudoviral neutralization assay, since we were struggling with how do we work or how do I identify labs that will be able to help us with live virus assays. What we found is that we needed to know the neutralization efficacy in order to find the best antibodies. The ELISA wasn't going to provide us with all the information that we needed. And so having the ability to run these assays in-house allowed us to do a lot more in characterizing and developing the best product that we wanted to move forward. So one of the first things that we did is we wanted to identify how much more potent can our recombinant antibody libraries get compared to the donors, the plasmas that the samples came from. And so for each of these pools, DP1, for example, is a pool of 2 donors, and the green represents the plasma, the light blue represents the recombinant antibody libraries that were developed by flow sorting the antigen, the RBD specific antibodies. And what you can see here is that each of the green line represents a couple of donors pulled together, and each of the blue lines represents the pool of the recombinant the B cells and the recombinant CHO cells that were created from those B cells. So you can see here that we're able to get an improvement by 100 or close to 1,000-fold more potent than the original plasma samples. And so this allowed us to see that we were getting the antibodies that we needed in order to move our product forward. So Giga 2050 is the final product that we developed by pooling together all of these individual recombinant library pools. Compared to IVIG, so in the black line, you'll see that there's no neutralization. And then we used 2 monoclonal antibodies, the SARS-CoV-2 monoclonal antibody CR3022, which was originally identified during the original SARS outbreak, but it's not able to effectively neutralize the virus for this SARS-CoV. The SARS-CoV-2 monoclonal SAD-S35, we demonstrated that our polyclonal antibody was almost as potent as this monoclonal antibody in neutralizing. This was the original SARS-CoV-2 strain, RVPs. One of the biggest limitations, as I mentioned, is having access to the neutralization assays and having live virus to be able to do that. We weren't able to work with live virus in our lab. We weren't able to find very many labs that were able to work with live virus. And so we wanted to have access to assays that would provide us with information about neutralization. But also we wanted to convince the FDA that our pseudoviral neutralization systems would be comparable to the live virus neutralization. And so we performed this assay comparing our Giga 2050 and some plasma samples using the pseudoviral neutralization versus the live virus. And we found that the results were almost perfectly correlated. We did run the live virus neutralization, and we demonstrated that our final product, was highly potent and was able to neutralize about 100-fold more potent than the plasma pools. And so we decided to move this product into manufacturing. One of the biggest questions that people will have about our product is that, can you demonstrate that you have consistency when you put each vial of cells into manufacturing? We can do that by looking at the performance of our final product comparing lot to lot. And so as you can see here, we have a number of development lots, tox lots, and then finally the 2 GMP runs that we performed. And we demonstrate that those runs and the results of the pseudoviral neutralization are overlapping. And so essentially the assay is showing us the products look the same, but it's also showing us that the assay is performing similarly. So the consistency of the assay is good and the consistency of our product is good. And so you can see that our product compared to the NIBSC standard, it's about 100-fold more potent than a plasma-derived hyperimmune used as the standard. It's also about as potent as the SAD-S35 monoclonal antibody, and then it's about 500 times as potent as a plasma-derived hyperimmune. With all this information, we wanted to go to the FDA and to demonstrate that we're ready for clinical studies, and so we submitted this as part of an IND package. And part of this, we wanted to use the RVPs for our lot release. And we were able to demonstrate that we're getting consistent results compared to our reference standard. And so we were able to move this into our final phases and submitting our IND for clinical studies. So this process was extremely fast. We had blood samples in our lab within weeks of the epidemic and the outbreaks happening after Mardi Gras in 2020. We had our libraries, our antibody libraries, ready to go within weeks of receiving those blood samples. We were transferring the research cell bank to the CDMO for making our master cell bank and getting ready for manufacturing. We did a lot of research and characterizing our polyclonal antibody and then developing assays for a lot release within the months after that. And then finally, our GMP lot was completed in December of that year. And we were able to enter into tox studies very quickly after that. We filed for our IND in January of 2021, and it was approved and ready for clinical studies soon after. Sadly, there were a number of monoclonal antibodies that were available at that same time, and we were having difficulty in recruiting for the study. So we didn't actually get to complete the phase 1 clinical study, but it was an exciting few months, and it demonstrated that we were able to isolate B cells, create this polyclonal antibody therapeutic, and get it through IND within a very short period of time. And we're looking forward to doing this again. And so I am very happy to present this work, and I'm looking forward to getting some questions from the audience. So thank you for listening.
I'd like to thank Integral Molecular for inviting me to speak about our experiences working with SARS-CoV-2 reporter-viral particles to support preclinical development of our subunit vaccine. This is the outline of my presentation. I'll touch on the remaining scientific challenges in COVID pandemic, talk about Sanofi COVID vaccine candidates, share details about pseudoneutralization assay development using pseudoviral particles, share with you some preclinical data in non-human primates looking at cross-neutralization, and then give you an update on subunit vaccine in clinical trials. It's safe to say that COVID changed our world a lot, and we're still continue living this reality. It has claimed estimated 15 million lives, and rapid development of COVID vaccines have been definitely a triumph of biomedical research. WHO reported as of May 2022 more than 300 COVID vaccines in preclinical and clinical development, out of which 8 COVID vaccines have been approved by WHO for global use. And global vaccination saved 20 million lives during the first year of vaccine rollout. However, initial goals for the COVID vaccination have been prevention of infection and transmission. However, this goal turned out to be not easily achievable. So waning immunity remains among the most important challenges of widely used COVID-19 vaccines. And other challenges definitely include the frequent emergence of SARS-CoV-2 variants and increased transmissibility and greater antibody escape. We observe as in front of our eyes, the COVID pandemic is transitioning from hyperacute to an endemic phase. We have learned that SARS-CoV-2 virus evolves at least 2 times faster than influenza, which was in the past considered to be the fastest evolving virus. These data on the slide, they are from global weekly sequencing posted by Los Alamos National Laboratories on SARS-CoV-2 variants over the period of 2 years from 2020 to 2022. And it slows very rapid evolution from Wuhan to Omicron. Mutation of Omicron variant seemed to happen even faster. This is the Omicron family tree that is already outdated. So late 2021, a highly transmissible Omicron emerged in Africa. Since then, in 4 months, the Omicron lineage rapidly splintered into at least 6 variants. And as I already said, this tree is already outdated because there are already additional variants that have been reported in the past month. So Omicron has substantial escape from neutralizing antibody and elicited by the non-Omicron variants. In this environment, frequent boosting for every emerging variant may not be really feasible. And the goal for COVID booster vaccine should really focus more on breadth of protection against variants and durability of the immune responses. Here, I want to talk about a Sanofi recombinant protein vaccine candidate. In Q1 2020, Sanofi joined collaboration with BARDA and GSK to develop recombinant subunit vaccine approach using baculovirus recombinant protein manufacturing platform together with GSK pandemic adjuvant AS03. Clinical development for the subunit vaccine started in September 2020. Since then, we've built an extensive clinical trial program to test 2 formulations, Wuhan D614 and Beta B1351, to develop vaccine that could help answer today's vaccination needs. So Wuhan and Beta spike protein sequence was modified with 2 mutations in S region, deletion of transmembrane domain, and addition of T4 fold-on trimerization domain to achieve pre-fusion stabilized trimer, and formulated, as already mentioned, with GSK AS03 adjuvant, which is a squalene in oil and water emulsion. The products produced in baculovirus platform require regular refrigeration, which is compatible with routine vaccine distribution requirements. A couple more details about the baculovirus expression system technology. It is an established commercial manufacturing platform for production of viral vaccines and gene therapy vectors. Multiple approved products have been generated using this platform, including Flublok, which is the influenza virus. And the platform offers really many advantages, including manufacturing speed, flexible product design, inherent safety and stability. It uses a single insect cell line, which allows for rapid plug and play, not as rapid as mRNA, but still rapid for the subunit for multiple products. SARS-CoV-2 spike protein sequence was introduced in baculovirus, which then used to infect insect cells. Following protein expression, the protein was purified according to standard techniques to a high purity. This slide shows really a rapid preclinical development that has been done to support progression of COVID subunit vaccine into clinic. The work came to my preclinical shop in April 2020, soon after Sanofi entered into collaboration with BARDA and GSK. And in a space of 4 months between April 2020 and September 2020, when a subunit candidate entered clinical trial, we have performed we have done the entire preclinical program, starting from the assessment of the selection of the candidates in animal studies, testing of functional antibody responses in several animal species, assessment of efficacy, looked at the Th1, Th2 cytokine responses, demonstrated safety in toxicology studies, and then carried assessment of cross-neutralization of variants of concern as they were emerging, and then supported submission of IND. This unprecedented speed would not be possible if we were not able to develop critical analytical tools to evaluate cellular and, most importantly, antibody responses. And functional neutralizing assay was definitely a key attribute critical for successful progression of a vaccine candidate to clinic. A few details on development of neutralization assay. At the beginning of the program, there was a discussion which assay to choose, live virus microneutralization or pseudovirus-based. Classical microneutralization assay clearly involves wild-type virus and obviously use need for BSL-3 facilities to perform this assay, which really brings serious constraints to the flexibility and throughput. So the decision was made to go with integral molecular RVPs with lentivirus backbone and GFP reporter gene. These pseudoviruses are replication deficient. They express on their surfaces S protein of SARS-CoV-2 variant and can infect 293 ACE2 cells overexpressing human ACE2 receptor. This platform greatly facilitated development of mix and read assay and allowed us for high throughput testing. You simply mix your diluted sera with RVPs and then put this mixture on 293 ACE2 cells for a single round of infection and measure the signal after 72 hours. Using the panel of human convalescent sera, we have performed concordance analysis between pseudoneutralization assay and wild-type virus microneutralization assay for Wuhan strain and showed a constant slight bias towards pseudoneutralization assay, constantly giving us a slightly higher titers. We also track the performance of convalescent control and RVPs only controls on top and on the bottom respectively on the slide, and have shown pretty stable performance. The data you're looking at here for the 6-month period. The account managers and technical team at Integral Molecular have been really knowledgeable, approachable, and responsive to our needs. They provided high quality RVPs and they always adhered to promised timelines, which was definitely greatly appreciated. So this slide shows you a list of some of the RVPs that we've used in our preclinical program. And Integral Molecular has developed actually pretty much all, I think, variants of concern and interest classified by CDC and WHO. And I think it's more than 70 at the moment. And this number continues to grow as the virus continues to evolve. And the pseudoneutralization assays are in line with the FDA recommendations for the vaccine developers. With that, I want to switch gears and show you our preclinical data generated in macaques. The data actually were recently published in Nature Communications. This data also were generated using pseudoneutralization assay. It was a key assay for this study. The question that we asked here was, what does a subunit booster do for neutralizing antibody titers when we look at the mRNA primed and versus protein subunit primed background. And so the study design is highlighted in this slide. So the macaques were primed with 2 doses of either a Sanofi unmodified vaccine, that's the vaccine we had early on, and then this program was stopped, or Sanofi protein subunit adjuvanted vaccine of Wuhan sequence. And then 7 months later, animals from both cohorts, mRNA and subunit, were boosted with either homologous formulation D614 on ASO3 adjuvant, or heterologous beta formulation with or without adjuvant, or also bivalent D614 and plus beta. Serum samples were collected from the animals after completion of prime immunization on day 35, wherever you see blood drops, and then before the boost and several times after the boost. So the first time we were assessed was pseudo-neutralizing antibody against the parenteral SARS-CoV-2. The mean neutralizing titers on day 35 after prime were similar in mRNA primed and in subunit primed. By 7 months post-prime, neutralizing titers dropped in both cohorts, and booster it dropped, and a booster with any tested formulation significantly increased parenteral titers as soon as 7 days post-boost. And then it reached plateau between day 14 and 28, and then we observed some decline over the period of 3 months. So the mean titer post-prime and even more so post-boost were higher than the titers seen in human convalescent sera. It's a graph on the right. So the next we assessed pseudo-neutralizing antibody titers against beta variant at different time points. After priming phase with Wuhan formulation on day 35, beta neutralizing titers were low to undetectable in mRNA primed cohort, and also low in subunit primed cohort. And 7 months post-prime, not surprisingly, beta titers dropped even further in both cohorts. A booster dose with all formulations, homologous and heterologous, as well as with or without adjuvant, induced beta neutralizing titers 7 days after injection in both cohorts. And this increase in beta neutralizing titers lasted over 3 months of follow-up post-booster. No adjuvant beta monovalent formulation, if you look at the red graph on the slide, was also capable of boosting neutralizing response, suggesting that adjuvant may not be actually as critical for the booster in comparison with primary vaccination. And this is the final slide of the data, looking at the all variants of concern, which were circulating at the time when the study was done: alpha, beta, gamma, delta, Omicron, the regional Omicron, and distant virus SARS-CoV-1. We're looking again in mRNA primed and in protein subunit primed, and all this data generated day 14 post-boost. And so we were able to detect cross-neutralizing antibody against all variants of concern. They were measurable in both cohorts. Interestingly, that and unexpectedly, actually, we did see neutralizing titers against SARS-CoV-1, even though it has at least 350 amino acid differences in S protein in comparison with SARS-CoV-2, but it was still neutralized at the same level as Omicron. And with that, I want to summarize the results I've shown you. We showed that in this study that 1 booster dose of adjuvanted D614 parenteral or B1351 beta in monovalent or bivalent formulations significantly boosted pre-existing neutralizing antibodies and elicited high and stable cross-neutralizing antibodies covering the 5 variants of concern, and unexpectedly also SARS-CoV-1 in primed animals. Interestingly, actually, the non-adjuvanted beta formulation also significantly boosted and broadened the neutralizing antibody responses. And this is the last point I want to make here, even though we did not assess it in our study, but there are a lot of published works about this, that another important consideration to keep in mind when you give the booster, it is the exposure, the interval between the exposure to infection or vaccination and a booster. Now I want to update you on our vaccine candidates' clinical development status. The subunit vaccine formulations are completing clinical trials, and here I'll just share the highlights of the most important studies. VATO-2 is the phase 2 dose finding study for monovalent D614 formulation as primary vaccination. The study was completed and the candidate was shown to be safe and immunogenic, and the data were published in Lancet. VATO-2 extended cohorts down below is done in individuals previously vaccinated with authorized mRNA or adenovirus vaccines, registered vaccines. We're testing here D614 beta or bivalent formulations, and the study is still ongoing, but we are seeing in preliminary results similar observations to what we've seen in primed non-human primates in terms of cross-neutralization. VATO-3 study is a primary vaccination study looking at 10 microgram dose of the D614 or bivalent and testing in naive or non-naive individuals. The study is still ongoing, estimated completion June 2023. And finally, VATO-13 study is looking at a D614 and beta as boosters. And it's actually a heterologous booster. So we're looking in individuals which were previously vaccinated with the Pfizer mRNA vaccine. And Sanofi, along with GSK, is moving forward with EMA registration for the beta monovalent vaccine in Europe as a booster. And I want to finish off with acknowledging the teams who have, which performed the work that I mentioned in the presentation today, COVID subunit, COVID subunit Sanofi team, GSK team. I want to acknowledge funding support from BARDA, our NIH collaborators, and I want to thank Integral Molecular for critical RVP reagents. Thank you.
Hayley, Sheila, Natalie, amazing work and some really wonderful looking data. So we thank you so much for sharing all of your hard work with us and all of your successes. We really appreciate that. To the audience, the Q&A is coming up in just a moment. So if you have questions for our panelists, don't hesitate to send them on in. We're going to try to get through as many of your questions as possible. All you need to do is type your question into the Ask a Question box and then hit submit. Okay, without any further ado, let's move on to the Q&A session and try to address all of your questions. Bear with us for just a moment as we make the transition into the Q&A.
All right, everyone. Thank you so much for joining us for this Q&A session. All of my panelists are here to answer all of your questions that have come in, and we have a bunch already. So ladies, you guys can open up your cameras and your microphones there. Thank you so much. And we'll start the Q&A. Hayley, the first question we have is going to go to you. An audience member would like to know, what target cell line do you recommend for SARS-CoV-2 RVPs?
Hi, Jeff. Thanks. So we established a monoclonal stable 293T cell line that overexpresses ACE2, and we send a free vial of these cells with every first RVP order, and these are the best target cells for SARS-CoV-2.
All righty, thank you for that. Sheila, we have a question for you. An audience member says, is an actual convalescent donor required, or could recombinant hyperimmune globulins be generated from sera from an immunized donor using technology described in your talk?
Yeah, so the important piece of making monoclonal or polyclonal antibodies is the gene sequence from the B cell. So a B cell is required. And so we need either a convalescent donor or a vaccinated donor. One of the really interesting parts of the technology is that we can find very rare antibodies. And so even if you had a donor who was weeks or even months or years out from an infection or vaccination, our technology can screen those millions of B cells for those single antibodies. So we do need B cells, we do need convalescent or vaccinated donors, but we can find those antibodies using this technology.
All right, thank you so much. Natalie, we have a question for you. An audience member would like to know, why did you choose RVPs for your preclinical work? Did you observe similar results using pseudovirus and live virus if you compared them?
Thank you, Jeff, for the question. I highlighted this a little bit in my talk. I can expand a little bit further. Yes, we have kind of had this discussion early on, which assay to choose, and we've decided to go with the pseudoneutralization assay for multiple reasons. One of them is really simplicity. It's a BSL-2 reagent. No need for BSL-3 lab, which the live virus definitely would require. So it's also we also have chosen GFP reporter-virus over luciferase, which was also available in neutralization particles, pseudovirus particles, I'm sorry, because that aligned with the other platforms, existing platforms at our company, and it is also a simpler assay. Luciferase-based pseudoviral particles require additional step where you would lyse the cells infected with the pseudoviruses and then add the substrate for luciferase. GFP does not require that, so it is literally mix and read assay. It's very, very simple. So as I mentioned, we were really on a very short timeline, studying the program a little bit later than many of our competitors, so we really could not go with the development of live virus assay. And there was also a very smart decision, because as the variants of concern started coming up, in pandemic, we were able to plug new pseudovirus particles in this assay very quickly, which would be much more difficult to do if we had live virus assay. And yes, we did compare pseudoneutralization assay and live virus particle assay using different serum samples. I shared in my presentation the panel, this comparison for the panel of convalescent sera. We've done it also for the sera from the immunized, from the vaccinees, and we've seen pretty comparable results. Sometimes pseudoneutralization assay gives us a little bit higher titers, but consistently the shift is consistent, but acceptable.
All righty, thank you so much for that. Hayley, another question for you. Jonathan would like to know, do the IAV RVPs both include HA and NA in the particles themselves?
Yes, the influenza A RVPs have both HA and NA on the pseudoviral surface.
All right, thank you for that. Sheila, question for you. Masha would like to know, has Giga 2050 been tested against the recent circulating variants of SARS-CoV-2?
Yes, so one of the great assets of working with RVPs and integral molecular is having access to those new sequences as they become available. And it's actually not too far behind. So, quite a short period of time lapses, and then the next variant is ready to go. So, we've been testing with each new variant, and with each new variant we test, we hope this is it. We're gonna finish this manuscript, and and then the next one comes out. So we have tested everything to date, and I think that maybe the most recent ones are the last ones, but we have a publication that is summarizing that data, and you can look forward to that, and then hopefully in the next few months.
Excellent. We look forward to that. Hopefully another strain won't come out before then.
I know. It's hard to know when to stop.
Yeah, exactly. Hayley, back to you. Another question. I'm sorry. Wait, Natalie, we had a question for you. Excuse me. An audience member would like to know, have you mapped any epitopes from some of the broadly neutralizing antibodies generated against SARS-CoV-1? If not, what are the technical obstacles?
Great question. It's not just against SARS-CoV-1. We were also interested to know about the the repertoire, of the map. Actually, we were interested to know the epitopes for the broadly neutralizing antibodies that we observed after the booster, because you definitely increase the breadth after you boost the previously primed animals. And we have actually confirmed this preclinical results in clinical trials as well. We are planning to do that. We haven't had a chance to do it yet. We have looked actually we've done extensive system serology analysis to look at the IgG subclasses and also at the Fc function in the antibody generated after different booster formulations, and we put this in a manuscript, which is actually submitted for publication right now. But epitope mapping, next step.
All righty, thank you for that. Hayley, we do have a question now for you. An audience member says, what protocols and customer support are available with an RVP order?
We have a lot of protocols on our website for infectivity assays, neutralization assays, how to passage and maintain the cells we send you, how to thaw them, to use them. We have everything available on our website. Just ask us for the link. But also we have a team of PhD scientists ready to jump on a call or answer any emails from any customers about how to use the RVPs. We can help you troubleshoot. We can take a look at your data. So there's definitely a lot of support and a lot of documents online to help you every step of the way.
Excellent. That's great to know. Thank you so much, Hayley. Sheila, back to you. Charu would like to know, was the FDA satisfied with receiving pseudovirus data once you had shown that there was good correlation, or did they push back and ask for live virus assays?
Yeah, so I don't believe that they even asked for the correlation, but it was something that we wanted to have in hand if the question did come up. They did not push back about having the actual virus assays as part of the lot release process. And to be honest, during the pandemic, it was difficult to get space in those in labs that had access to live virus, since it was a BSL-3 lab that those assays need to run in. So we did not have any pushback from the FDA, which is it was really an amazing process, and especially being able to use the pseudoviral neutralization. Hopefully, this establishes the path for the next products using the next pseudoviral assays.
All right, excellent. Sheila, I'm sorry, Natalie, excuse me, over to you. Tilahuna asks, are you concerned that non-live subunit vaccines are considered to be poor inducers of CMI? CMI is considered superior to protection against viruses than antibody response. Have you looked at CMI responses?
Great question. We are not concerned because we did look at the CMI responses. We have observed them. We do elicit T cell responses. In fact, it was requirement from FDA early on for all vaccine developers that we would provide data showing that we have Th1, Th2 balanced response and not Th2 skewed, because at the beginning of pandemic, there was, as companies were coming up with the vaccines, there was a concern that we might see the disease enhancement with Th2 skewed type of response, which we haven't observed so far. But to be honest, all the vaccines, definitely all mRNA vaccines, they show very strong Th1 bias. Our subunit vaccine was strong adjuvant. Majority of the actually S protein is the part of in the vaccine. So there are plenty of the T cell epitopes there. That's actually one of the reasons why we did not choose receptor binding domain, which would be a much narrower epitope and a more focused for elicitation of B cell responses. We wanted to have broader epitopes, and we wanted to have to make sure that the T cells will be elicited as well. So yes, we did test for the T cell responses. We did see a very balanced response, definitely not Th2 skewed. We did look for memory B cells as well, which is a little bit different kind of a question, but we do have actually all those responses elicited with our vaccine. So we are not concerned.
All right, good to know. Thank you so much. Hayley, back to you. Masha has a question for you. She'd like to know, are the RVPs produced in HEK293 cells? What process is followed to concentrate the RVP post-production?
Yes, our production cells are HEK293 T cells. And the RVPs in our catalog are not concentrated at the moment. We tend to get high titers for all of the variants and most other RVPs without concentration, but we do offer concentration as a custom option if you have specific bioassay needs, or if you have a custom project that has more challenging viral entry and fusion proteins. We can definitely offer concentrating, but our off-the-shelf RVPs don't need to be concentrated.
All right, great to know. Natalie, back to you. We have a couple questions here for you, which I just scrolled past, so give me 1 second here. Oh, here we go. An audience member would like to know, why did you choose RVPs for preclinical work? Did you observe oh, wait, I'm sorry. I think I already asked you that one.
Yes,
My bad. Sorry about that. Here we go. Have you mapped, nope, I did answer that one too. My bad. Apparently it's not keeping my things here. Here we go. When you're looking at immune responses from large numbers of patients in clinical trials, how many variants will you need to assess using large number of patient samples?
Well, it depends which clinical trial you are assessing samples in, because in phase 2, pretty much my answer would be you are expected to assess response to all circulating strains. So if in earlier part of pandemic, when we had alpha, beta, gamma, delta circulating, you are expected to assess your immune response to the strain which is in your vaccine, obviously, right? So homologous to what you immunize with, and then to all the circulating strains. So then it was the later phases of clinical development in phase 3. For example, we were expected to assess to homologous what's in your vaccine and what is circulating, which at the time was already all the variations of Omicron, B1, B2, and 4 and 5.
All right, excellent. Hayley, back to you. Let's remember a pretty important question. Would like to know, what QC work do you do on RVPs?
So for every lot we produce and every variant we produce, we test the infectivity of the RVPs. We test the neutralization against antibodies that we have generated and commercially available antibodies. We also test for mycoplasma, and we only release a lot if it passes everything. And then in addition, some customers ask us to do extra QC. They may have a different plate layout. They may have extra testing they would like done, like sterility testing or endotoxin. So we have standard QC that is very strict, and then we can add on extra QC if it's required.
All right, excellent. Natalie, back to you. Charu has a question, and she'd like to know, were you required to produce neutralization data against the different variants as well?
Yeah, that's a good question. Pretty much, again, the answer is very simple. You are expected to, as we were not the first ones to go into with our vaccine development, right? So by the time we came to clinical development, there were a lot of other companies that were already in clinical development. So when you come, and with the pandemic, actually all the things were set as we were going in a way. So when we were coming in, there was expectation, and we were submitting our data to regulatory agencies. There was expectation from regulatory agencies that we provide the same data as the others already provided. So, and the others provide what is in, the data against all circulating strains. So, pretty much the answer is, and of course, you are, as a vaccine developer, you do want to know what your vaccine is capable of doing and against the circulating strains. It wouldn't be interesting to know that your vaccine is, whatever it's doing, the immunogenicity and protection against a strain which was circulating at the beginning of pandemic and no longer is in circulation and prevalent, right? So pretty much you are, there's internal and external expectations that you will show you will test your vaccine against everything that matters and pretty much everything that is prevalent in circulation at the moment.
All right, great. Hayley, simple, straightforward question, but also an important one. Can RVPs be used in clinical trial setting?
Yes, absolutely. They have been validated for use in clinical trials by some very large CROs that are our customers for SARS-CoV-2. And then Takeda, who I mentioned in my talk, they used our Zika RVPs in their phase 1 clinical trial. They used our dengue RVPs in their phase 1, 2, and 3 clinical trials. And because we have customers that test thousands and thousands of sera samples, we are able to produce really large lots of RVPs, liters of RVPs, so that these customers don't have to do bridging studies.
All right. And Hayley, stay with me for 1 second. I got one more for you. Do you recommend GFP or luciferase RVPs?
We get this question a lot. I think half of our customers use luciferase and half use GFP. So there's really no preference. I think it comes down to what equipment you have in your lab and what you want the readout to be. So for luciferase, you get a population average result and you use a plate reader. For GFP, you can do single cell analysis by flow cytometry. So it depends what equipment is available and it depends what you want your readout to be, but they both work equally well.
Right. Thank you for that. And I'm just scrolling through some of the answers here, questions, see if we got any more. It looks like, Hayley, we got one more for you. It looks like Sick June wants to know, how do you concentrate and store pseudoviruses?
So we store them at -70 to -80. And when they are thawed, they're thawed in a water bath very quickly and stored on ice to then be used within 30 minutes or an hour. In terms of concentrating, there was another question earlier about this. We don't concentrate them as standard practice, but we can for a custom project. So if you're interested in that, just get in touch with us.
All righty. Thank you so much. And cycling through all the questions, I think we've come to the end of our webinar. I don't really see any more questions unless you guys see any that I'm missing here, but I don't think so. So
I see one, Jeff.
Oh, yeah. Okay.
It says from Thomas. Have you
okay.
noticed any large differences in yields when switching between different pseudotypes? Have you had to significantly modify these envelopes to get the desired titers? What types of modifications are made? So yes, often with these coronavirus variants, they have so many new different mutations that it's as if we're making different viruses each time, especially with Omicron and all of its lineages. So we definitely see different titers coming out of each production. We don't modify the envelope protein though, not for SARS-2. We use the native full-length spike protein, same for dengue, Zika, Ebola, and Marburg. However, if we come across a tricky variant or if a customer gives us a sequence that is tricky to work with, we do protein engineering here at Integral to improve titers or to make these RVPs possible. So we do sometimes make modifications. It's not common though. If an RVP is hard to produce, we will often change the production conditions first before genetic engineering. But yeah, we have many ways to tackle these pseudoviruses.
All righty. Thank you so much for that, Hayley. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the Gen website. That's at genengnews.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any of your friends and colleagues, which we always recommend. I'd like to thank Hayley, Sheila, and Natalie again for their very informative presentations. And I'd like to thank you, the audience, for your attention and very thoughtful questions. And a very special thanks to Integral Molecular for sponsoring this webinar. Hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now. Everyone stay safe and healthy.